All right. Thank you very much for coming. My name is Dr. Julie Dumas. I'm a professor of psychiatry at the University of Vermont. I'm the Vermont principal investigator for the CHAMP study. Let me introduce my colleague, Dr. Paul Newhouse, also professor of psychiatry and Jim Turner Chair in Cognitive Disorders at Vanderbilt University. He's the principal investigator at the Vanderbilt University site. Some folks here participated at UVM, some folks at Vanderbilt, and some folks did both, which I'll talk about as we go. We're going to do tonight is Paul and I, or Dr. Newhouse and I, are going to tag team the presentation. We'll talk about some of remind you of the sort of the science behind it, the logic of what we're studying. I'll present some of the results, and we'll talk about next steps. So let me get the slide sharing going. Hmm. Okay, let me get it in business. Okay, oops. All right, is that working, Paul? Am I sharing this? All right. So I'm going to turn it over to Dr. Newhouse, and he's going to start off with the intro and the logic about why we're here and how we got to this point and where we're going. And I guess I'll do where we're going next. So take it away, Paul.
Thank you, Julie. Hopefully everyone can hear me. Why don't we go to the next slide? So I hardly need to be explaining menopause to a group of women
Sorry.
who've already experienced this, but just to refresh our memory collectively and also to get us on the same page, we've been interested in menopause as a risk factor for women in late life and their brain health for a long time. And Dr. Dumas and I have collectively had almost 25 years in studying what happens after menopause, what happens with hormones, and what happens to the brain. And so we have been very interested in this menopausal process to remind everyone during a woman's reproductive years, she has a normal cycle of a series of sex hormones which are related to reproductive function. This cycling is driven by brain mechanisms and involves structures in the brain, such as the hypothalamus and the pituitary gland, and also, of course, the principal gonad in women, which is the ovary. And the ovary responds to the signals that it gets, and you get the left side of this picture with a rhythmic monthly cycle of a series of hormones which produce both the secretion or the production of eggs from the ovaries and also changes to the lining of the uterus, followed by the ending of that process with menstruation. During the perimenopausal interval, these rhythms become what we would say in medicine dysrhythmic, or they lose their rhythmicity, and we get a more irregular cycling period. Menstrual periods get longer and more erratic, and fertility, of course, is reduced significantly. This settles down after a period of several years, and it can be very individually variable how long that period is, into a postmenopausal state when the cycling essentially stops and we get fixed values of these sex hormones, where estrogen is very low, where progesterone is very low, and other signaling molecules like FSH or LH actually paradoxically become higher. And so that leads us to this sort of steady state process that proceeds through the rest of a woman's life. Next slide. So the ovaries stop producing estradiol and progesterone at menopause. They actually still do, the ovaries still do produce a little bit of testosterone, interestingly. Pregnancy, of course, can no longer occur because the egg supply in the ovaries becomes either exhausted or non-functional. And estrone, which is not estrogen, or it's not the normal estrogen we think of, which is we call estradiol or E2. Estrone, which is a weaker form of estrogen, becomes the predominant circulating estrogen and does not vary over time, but is rather steady. Next slide. So we've been interested, as I mentioned earlier, in the idea that menopause is a kind of big biological event that can't help but have impact on a woman's biology and on her physiology and, of course, her brain. And so we have suspected, and there are many other investigators who have suspected for many years, that this natural condition has implications for cognition and brain aging. Next slide. So we normally see a number of sort of neurological and psychological impacts of menopause. We see a more disturbed sleep. We see disruptions in thermoregulation, such as hot flashes. We have the autonomic nervous system becomes unruly, as one investigator put it. We can see changes in mood in some people who might not have had mood changes before. And then we can see changes in cognitive performance with changes in what we call episodic memory or working memory and attention. But studies have generally indicated that for many women, maybe even most women, those changes don't necessarily persist into the long postmenopausal period. So there is some cognitive disruption in that menopause transition or perimenopausal zone, but those generally recover after menopause completes the process. Next slide. But unfortunately, what we also noticed just from epidemiologic data is that women seem to be at elevated lifetime risk for Alzheimer's disease. And you can see on this left slide from data that's now almost 20 years old, that women have a substantially higher risk of lifetime risk of Alzheimer's disease. And that persists even past age 65, which is in this slide. And if so we don't know fully the reasons for that, but that's part of the rationale for the CHAMP study. And on the on the right side of the slide, we have a sort of follow-up trajectory of individuals in a long-running cohort study called ADNI. And we notice that women in green versus men in sort of maroon color tend to show faster or greater progression of cognitive impairment over time compared to men. And although we don't have a slide of this, myself and other investigators here at Vanderbilt have suggested that one of the reasons for this could be that women spread these abnormal proteins that we see in Alzheimer's disease more efficiently, which may be not a good thing for Alzheimer's disease. But there are many particular or potential links to this, and one of them is menopause, and we wanted to further understand that. Next slide. And menopause is an interesting concept also from an aging perspective, because if we look back all the way to 1900, the average life expectancy for a woman was not a whole lot different than the age of menopause. But by now, of course, we've increased dramatically the life expectancy for both women and men. And now, for example, the average life expectancy for a woman in 2025 is 81. And I suspect, in fact, I know that for a woman who's reached the age of menopause, that life expectancy is closer to 85 now. If you've already survived to menopause, your life expectancy is probably around 85 for the average woman. So in a sense, women have outlived their reproductive life, and we have to sort of understand what the implications of that are. Next slide. And one of the implications is that there are changes in a whole bunch of neurotransmitter systems that seem to be affected by estrogen. And we have been particularly interested in one of these systems, which is the one in red on this slide, acetylcholine, which is located, the centers for that are located in the base of the brain in something we call the basal forebrain. There are other transmitters, though, that are also influenced by estrogen. I'm not going to talk about them much today, but they're dopamine, which you've probably heard of, serotonin, which is linked to mood, and even norepinephrine or noradrenaline, which may be linked to things like arousal. Next slide. The cholinergic system is particularly interesting to us because it affects performance on tasks that require attention. And attention is the front end of memory. If you don't attend to something, you don't remember it. And so we believe that different structures in the brain, the frontal part of the brain, or what we call the hippocampus, which is in the temporal lobe, are important and are modulated by this cholinergic system, particularly when there's a lot of need for attention. Next slide. One of the things we know about this cholinergic system is that aging and Alzheimer's disease can lead to a deterioration in this system. We know from data from many years ago now that the volume of this structure tends to get smaller as we age. That's the left side of this slide. You can see that these are very tiny structures. The brain image in the bottom right-hand side of the slide in those little colored areas shows you how tiny these structures are compared to the rest of the brain, which is huge by comparison. But yet they send an enormous number of nerve cells and nerve processes following those blue arrows on the upper part of the right side of this slide. They send all these very long axons all the way through the brain to innervate thousands, maybe millions of cells through the brain. And so they have a disproportionate impact on how our brains function. Next slide. All right, so I'm going to now stop and then turn it back over to my colleague, Dr. Dumas, who will take you through how we study this and why we wanted to look at postmenopausal women.
All right. Paul was just talking about the cholinergic system. And so how are we going to study the integrity of the cholinergic system in humans? So if we had a little rat or an animal model, we might be able to lesion the cholinergic system and see how the brain works without it, but we can't do that to humans. So what we do in our studies, and you guys participated in this, is we use a medication that temporarily affects cholinergic system functioning. Then we measure cognition, and then people recover and they go about their day and everything is back to normal. So it's called a transient neurochemical lesion model. That's a lot of words. And so the medication we used in this particular study was called mecamylamine, and we used mecamylamine compared to placebo, and I'll talk more about to remind you what that looked like. And this is a medication that temporarily sort of blocks cholinergic receptors, a specific kind, and we think might change cognition for a period of time. So that's what we were measuring in the CHAMP study. And there's another part of the study where we use a different medication. We can talk about that a little bit later. But Dr. Newhouse has been doing this since, I don't know, since the brain was invented. But you and I have been working on this for about 25 years in these sorts of models to use these cholinergic drugs to see how they affect brain functioning in middle-aged women. And so here are some results from a prior study, not your study. And I would always say to Dr. New, say, you know what, this doesn't even work. It doesn't even impair cognition because when you take the average, there's sort of this is a study of the mecamylamine compared to placebo, and on average, there's no difference between performance. But if you look at individuals, some people actually perform better during mecamylamine. This is a memory task and scores above the zero or better performance, and some people are impaired, which is exactly what we thought would happen. So we sort of said, no, this is interesting. There's a lot of individual differences here. And can we tell anything about these people who maybe have this compensation or might perform better during the medication, which is not what we thought would happen, or the people who are doing the thing we would expect and have this sort of impairment for the medication? So we took that model and we looked at it in menopause, and this is a lot of data. I just want to make a couple of points. And we did this in postmenopausal women where we actually treated women with 3 months of estrogen as well as 3 months of placebo. And then after those 3 months, we did these cholinergic challenges. What we found is that if you've had estrogen for 3 months, the cholinergic impairment is less. So having estrogen on board supports cholinergic system functioning, and even the medication that impairs cognition sort of doesn't work as well, or doesn't. Estrogen is protective for cholinergic challenge or cholinergic drug in this case. We've also shown that this is, oops, excuse me, wrong way. That this is the case for this is works sort of estrogen works better with protecting cholinergic systems for younger postmenopausal women compared to older postmenopausal women. I'm not going to talk about that too much unless we get into the question and answers, but we've shown that there's this critical period for when estrogen is protective for cholinergic system functioning. That made us think, so what's going on at menopause or close to the menopause transition that might protect the brain for in future aging, or maybe is making it vulnerable. And that is leads to CHAMP. And so that's all lot of in 2 slides, a summary of many years of work of Dr. Newhouse and myself. And so that leads to CHAMP. And we are interested in sort of how are all of the things that go on at menopause for a woman, how do they sort of set a woman up for healthy or normal cognitive aging, or perhaps are at risk for dementia? So we measured, and we'll go over what all of the things we measured in a moment. And we sort of wanted to look at sort of how these things sort of load on what are protective factors and what are factors that might increase risk for pathological aging. So just to remind you what I'm sure you all know, so that we aim to have 120 cognitively normal postmenopausal women aged 50 to 70 at both sites at Vanderbilt and the University of Vermont. As you'll recall, you came in for 2 MRI study days where we had the medication, mecamylamine or placebo. So I'll show a timeline of that in a second. You took a pill in the morning and you waited 2 hours and you went down and did the MRI. We also did some structural MRIs. I'll show some data. Another day you came to the hospitals for a PET scan, and we were able to look at amyloid. And amyloid are the proteins that show up in Alzheimer's disease that are sort of the abnormal proteins. These are sort of the garbage that's left behind, and these are things we don't want to see. But we were looking for that with the PET scan. Many of you participated in the lumbar puncture, which we greatly appreciate, the spinal tap, and we were able to take some spinal fluid and look for biomarkers for Alzheimer's disease. So some things that might indicate in the cerebrospinal fluid that there was amyloid or another protein called tau in the brain. You wore the watch, the actigraph thing for a week, so we were able to measure sleep and activity. We also did a bunch of cognitive measures every time we saw you. We took blood on the first challenge day for the biomarkers that are in the blood, so we can see if can we get better measures. The blood measures actually, when we first got the grant, the blood measures weren't technically advanced. They like hadn't invented them yet, and as the grant took a while to the study to go on, that the science got better over time. We were able to measure these proteins in the blood, and that's a really cool advance. And Dr. Newhouse can talk more about that, how that's out in the world. So we were able to do that with this study. You may recall a reproductive history questionnaire, like how many kids did you have? Did you breastfeed? All kinds of things like that. And then some folks were able to participate in another PET session. And so this is the one that happened at Vanderbilt only. And we had a radioligand, a PET, one of you got injected with a ligand that actually stuck to cholinergic neurons. So you're able to see sort of where in the brain the cholinergic neurons are. And then we look at how that relates to all the things that we measure as well. We didn't have the ability to do that at Vermont, so we actually sent many of the Vermont women to Vanderbilt to participate in this part of the study. And there was also an EEG where you wore the funny cap and we measured sort of brain activity. So that's what you guys did in the study. And here are the results, or not the results, but some of the demographic data to tell you about who's in the study. So we had 114 postmenopausal women, on average age about 60 years old. People had a little bit more than college education, and years since menopause was about 10 in this group. We did some genetics for a gene that is related to risk for Alzheimer's disease, and there's one particular gene that sort of increases risk, and some others that might be protective. And so we saw some different people had different genetics, which is we would expect. During our cognitive screening, just to let you know that everyone was cognitively normal and healthy, people might have heard of the Montreal Cognitive Assessment these days, the MoCA. It's in the news. Everyone did very good on the MoCA. People were not depressed on a measure of depression. I think our depression score was like over 10, but everyone, not depressed, not anxious. Again, our anxiety cutoff was about 15, so not anxious. People were cognitively doing very well, way above average, and had good functioning working memory. So everyone was cognitively normal and healthy as we were expecting. Oops. Okay, so just to remind you, when you came into those MRI study days, you would arrive at the research center at either institution. We then give you some pills to take by mouth, and then we wait 2 hours, and then we take you down to MRI and do the MRI and the cognitive testing, bring you back, give you lunch, and send you off. Hopefully, you're feeling back to normal. So this is the lesion model that we like to the transient lesion model. So we give you the medication. It takes 2 hours to be working in the brain, and then we measure cognition after that. And here is what I will like to remind you of the NBAC task, the letter task. And we're still using this in other studies, and nobody likes this, no matter how old you are and no matter how much we train you. This is everybody's least favorite thing. So this is the task that you did during the MRI, where you saw letters appear on the computer screen one at a time, and your task was to decide whether the letter matched according to a certain rule. And we told you what the rules were, and the rules changed over time. So the first condition is the zero back. So you might say, okay, the matching letter is X. Whenever you've seen X, hit the match button and hit the mismatch button for all other answers. So you might see A, B, X, C, D, X. So you're just looking for X, pretty low working memory load. Then we say, okay, the condition is now the one back. So every time you see if the current letter matches the letter one back in the sequence, then you made a match response, but everything else is a mismatch. So you might see, excuse me, A, B, C, C. That second C is a match because it matches the letter right before. D, E, E. So that's a sort of, you're looking for two in a row of the same item. Then the two back, so there's sort of one intervening item between the missing letter, the matching letters, and then everyone's favorite is the three back. So you have to keep sort of four pieces of information in memory, then get another piece of information, update memory, and sort of shift your mind as you go along the letters. So the point of this is that it is hard. So as the n increases from 1 to 2 to 3, you use more brain to do the task. And so what we're looking is to see how the medication affects the increase in activation as the task gets harder. So let me show you that. I'm not sure these are your brains, but this is an example of what the task looks like. So this is your brain on the NBAC. And so what this orange colors indicate is that as the task is getting harder, your brain, more blood is flowing to these areas in the brain to sort of help you perform the task. And so this is an increasing working memory load as the task gets harder. So now let me show you some of your data. So what happens during the mecamylamine? So the orange means that there's more brain activation as the task is getting harder. And now here's what happens when we use mecamylamine. So now the brain is sort of, it's working, not as less hard. It's actually, there was more activation during placebo. So this is weird. This is not what we expected. This is not what we predicted when we wrote the grant. We thought it might be a little different, or we might see some orange in the front. So let me orient you. Sorry, your eyes are here, and this is the back of the brain. Down here, your eyes are here, and this is the back. So we thought we might see this orange, this increased activation, and then the blue back here, but that's not what we've seen. So this is interesting. I love when we get a result that we didn't expect. So now what does that mean? How do we think about this? How do we put this all together? And I'm going to show you what we're going to do with it, which we haven't done yet. So these are data from CHAMP. This is the sort of looking at the challenge results and sort of seeing how people did when they had the medication. And so this is a memory task. Remember, you may recall we read to you a list of 16 words and you asked you to try to remember them. And this is this is the total number of words you remembered. And So what we did was we took the number you remember during mecamylamine and subtracted the number you remember during placebo, and this is the difference here. So zero means that you got on zero on the on right here indicates that you were not different on mecamylamine and placebo. The little dots above those people performed better during mecamylamine, and the majority of people were challenged during the mecamylamine. So that's exactly what we would expect. But we're interested to see, what is the difference between these folks? So what we're gonna do next is we're gonna see who the folks who are up here who are performing better during the drug compared to the folks who are experienced with the expected challenge. We're gonna see how these things relate to the biomarkers or the other measures that we have that I'm going to talk about next. So that's the next step, and we'll also go back to that brain imaging picture and see if we can make these two groups and see if that changes the increased activation or the decreased activation during the medication challenge. So let me just show you some of the other relationships that we found. We are still working with the statistician to put all of this together and make one story, but as you may recall, there's so many measures that we took. So we're just going to try to talk about some of them that we have. So one thing that I think is interesting is that if we look at menopause symptoms, so you answered a questionnaire about menopause symptoms in the last 4 weeks, and most people were 10 years since menopause, right? So that's about, I think some of these things persist. What we found is that folks who reported symptoms like hot flashes and night sweats also reported poor sleep. They didn't think they were as good cognitively as they were 5 years ago. They reported some mood changes, and a little bit of anxiety. So all these things were also, as the menopause symptoms increased, so did all these other things. So that's sort of an interesting relationship or sort of thing, symptoms hanging together. That's sort of having all these symptoms, or that could be because maybe people notice their symptoms more. So I'm in tune to what my body is feeling, and that might be one of the explanations for this relationship. Here's an example of the sleep data. So the sleep are from the Actigraph, the watch that you wore for 7 days. We're able to put it through some computer software and come out with a measure called sleep efficiency. I think it's sort of like how long you're actually sleeping versus how long you're in bed, or some calculation like that. And we just graphed it across the age range. So our folks, as you know, are 50 to 70. You can see that sleep efficiency increases as people are older, for all the older groups or the older folks. So that's really interesting. We would expect, and I think other data show the opposite, that sort of as you age, your sleep declines. And again, this isn't longitudinal data. We didn't follow people as they age. These are just like snapshot of one time. But it's sort of not what we would expect. So this is interesting, and we'll dig into it and see what we can learn about this. Here's a lot of pictures, and this is some of the data of the cholinergic PET. So this is again the PET scan that we could only do at Vanderbilt, that folks traveled for, and the Vanderbilt folks did there as well. And this lets it what we're seeing here in these pictures, the bright red is where the tag is lighting up the cholinergic system, or where there are cholinergic neurons in the brain in these bright spaces. And so we're able to measure sort of how much that red stuff is there, and show relationships with some of our other measures. So this is a relationship to the binding, the cholinergic binding, as well as age. So you can see that as the older groups, there is less cholinergic binding, not groups, the older people. So that's exactly what we would expect. These things change with age. It's very normal. So this sort of is a sanity check of like, yes, this is behaving as we think it might. Down here, we have a measure. This is on the y-axis. We have the measure of the cholinergic binding with a part of the brain that is that Paul showed at the very beginning, that really tiny little colored thing on the brain, the nucleus basalis. And that's one of the cholinergic nuclei. And so as that volume gets bigger, there's more cholinergic binding. Again, another sanity check that as the brain area gets larger, there's more binding. That's exactly what we would expect. And then there's a cognitive measure. So there's less binding as performance becomes actually switching time. I guess performance comes worse, or I don't know. So now I'm confused about this, but I thought this meant that less binding with less good performance, but it might be the opposite now that I'm thinking about it out loud. But anyway, so there's interesting relationships between the cholinergic system and age and performance and other brain structures that we're still working on and still digging into. I'm going to not switch gears, but another piece of the data. These are the biomarkers from the CSF as well as the blood. And these are the things we didn't have available when we started the study. And as science continued, as we were doing the work, we were able to look at these things at the end. I think the data from the CSF didn't really show anything interesting that we've looked at so far. We're still looking at it. And there are a number of biomarkers that we were looking for. So let's see if we can see these Alzheimer's disease proteins, and they're called beta amyloid and tau. Let's see if we can see these in the brain. And actually, just written down here, we really didn't, not that people didn't have them, but they weren't related to age, they weren't related to brain structure, they weren't related to function. They were just very low levels and not related to anything. So we found very low levels of these proteins. But what we did find is some other protein, other not protein, other proteins, other molecules that aren't just maybe not just, but might just be related to aging or in general. So something called GFAP sort of increased with age, as well as something called NFL or neurofilament light also increase with age. And these are things, this sort of makes me think, okay, so this is again one of these sanity checks. Things are these markers of general brain aging are increasing with aging. So that sounds again a sanity check, but I'm happy to see that there's not this increase in Alzheimer's markers. So that is interesting and in this sample. This is one of the, this is a molecule called p-tau 217. I'm going to let Dr. Newhouse talk about it when he gets the floor back in terms of, I think this is might be approved for looking at Alzheimer's disease or sort of a biomarker of Alzheimer's disease that might be able, they run them in labs, special labs. And this is sort of people are getting this test run. And we have it here, and there's just no relationship of this molecule with age in this sample. So, our levels are just really low here, and this is just not showing us that there's a lot of p-tau 217 in our sample here. So what is next? That was a quick run through of the data we have so far. But what we are continuing to do is to look at sort of what are the measures in CHAMP that might be related to these biomarkers, to the blood measures that I showed you. Maybe the CSF hasn't shown us anything yet, but maybe we are still looking. Some of the PET data, again, that we didn't really find in the amyloid PET, we didn't find the proteins that we were looking for, but there might be some other relationships that we can look at. And what we do want to do is I showed you those data with the people who performed better with mecamylamine compared to the people who had the performance we expected. We want to see if this has any relationship to folks who might, to these Alzheimer's biomarkers. Are these people, are these are different based on performance during the medication, but can we say some are at risk and some are resilient? I'm not sure we can say that. So we're going to look and see how these measures relate to the other all of the data that we've collected. So we are continuing to examine cholinergic PET so that we looked at some of the data. I think the folks at Vanderbilt have looked at the EEG data. I just don't know all about it so far. They're finding some interesting relationships. Sleep activity and reproductive history, we think all of these things might sort of tell us a little bit about how these brain data, the cholinergic data, and the biomarkers relate to sort of what these individual difference factors, which is how we got into this business in the beginning. And then, of course, Dr. Newhouse and I want to do this, not the entire thing, but CHAMP Part 2. So how has, when we started the study in 2019, now it's, I don't know what year this is, 7 years later, and maybe if we get some more funding from the National Institute on Aging, how's the last 5 or 10 years changed any of these measures? So we are interested in sort of looking, calling you all back in a few years. Hopefully we'll get funded again and continue to see how these things are changing in our sample that we've done a lot of characterization with. All right, is Kim or team on the call? I'm sorry, I can't see you because I have my Zoom set up funny. And
Hi.
hi, do you want to unmute and introduce yourself?
Yes. Can you hear me?
Yes.
Great. Hi, I'm Kim Albert. I'm a neuroscientist here at the Center for Cognitive Medicine at Vanderbilt. And Brianka, who is the coordinator for this study, is also on the call here. So this is a study that we've been running relatively newly, about a year, here at Vanderbilt. Some of you have actually already participated in this study. And it's very similar to the CHAMP study, but we're looking at slightly different questions. And so, as kind of has already been discussed, there are some older adults who notice changes in their memory, but that these changes don't show up on clinical tests. And these subtle changes may be early markers of brain changes occurring with aging or indicators of dementia risk. And so we're looking at how these subjective changes are related to cognitive performance and changes in brain functioning. So this study, like I said, is very similar to CHAMP. It uses electroencephalography or EEG to measure brain activity during cognitive tasks. And EEG is essentially a little net of sensors that sit on your scalp and measure the brain's electrical activity. And so this is non-invasive and non-painful. And really what we're hoping to understand is how these changes in the brain are happening, and this might
Sorry.
help us improve future screening for dementia risk and prevention of dementia. So the study includes 4 visits. There's a screening visit, a practice visit, and 2 study visits similar to CHAMP. And the study visits do include a study drug challenge, testing, and the EEG session. To be eligible for this study, you have to be over the age of 55, but no neurological disease or current mood disorder, non-smoking, and not having taken ovarian hormones in the last year. Also, this study is only taking place at Vanderbilt, so you need to be able to come to Vanderbilt for the study. If you're interested in helping us out and participating, you can contact Brianka. This is Brianka Renfro, and her email and phone number are here on the screen right now.
Can
Thanks.
somebody paste that in the chat? Because I'm sharing my screen. I don't have, I'm like, I can't. That's why I keep messing things up. So
I can do
somebody,
that,
Megan or
yeah.
Kim, can you paste that in the chat so folks can see it or can think about it or grab it
Yep.
they want to. Um, thanks, Kim. Sorry, I didn't want. Okay. Are you all set? Can I go?
Yeah, you can go ahead.
Okay, great. All right. Thank you. All right. Dr. Newhouse, you want to wrap up the sort of why we are here and where we're going story?
Right. So thank you, Dr. Dumas. Wonderful presentation. So I want to emphasize to our amazing participants in this project that you now represent the most well characterized individuals in the world now for at your age for these types of brain measures, biological markers, EEG. There has been no group of women anywhere on the planet that has had this unique sort of deep measurement of all these characteristics. And why that's important, if you could just pop the red arrow on, Julie, is that we're very interested in sort of understanding what's happening in this sort of middle phase of life when some people may be starting to develop symptoms or rather biological changes that could later lead to symptoms of memory loss. And so this sort of cascade of events that occurs in Alzheimer's disease, we're interested in understanding this so we can make recommendations about potential prevention interventions down the line. Next slide. So I'm often asked, well, what can we do to reduce our overall risk for Alzheimer's disease? And so I like to sort of have this kind of wheel that's illustrated in this figure over on the right side of the slide, which includes sort of your genetics, which you can't do anything about, aging, which happens to all of us, whether we want it or not. But there are many other things that we can do to reduce our risk of brain damage or brain aging effects. We can reduce our risk of inflammation by controlling our weight, by exercising, by controlling our blood pressure. And what's good for the heart is good for the brain. We have evidence that infections, particularly chronic infections, can produce brain damage. So for example, there's recent data that shows that getting a shingles vaccine actually reduces the risk of developing dementia years later. So vaccines may actually be protective. Certain foods are associated with reduced risk of dementia, at least on a population level. Things like the Mediterranean diet, something that's high in vegetables, fruits, legumes, beans, fish, and lower in meats and dairy products and sugars, seems to pay dividends to reduce our risk of Alzheimer's disease and dementia. And so intellectual engagement seems to be very important in maintaining our brain's ability to scaffold itself to support aging. The brain has many redundant mechanisms there, and we want to encourage the development of sort of compensatory strategies, just like we have to exercise more when we age just to maintain our strength. We have to intellectually engage more as we age to maintain the cognitive function we already have. And so there are many things we can do. If we have a positive family history of Alzheimer's disease or a positive risk for a genetic risk factor for Alzheimer's disease, like ApoE4, we particularly want to take action on these what we're calling modifiable risk strategies. Blood pressure seems particularly important, and having low enough blood pressure is really critical because there is clear evidence that high blood pressure produces long-term brain damage. Next slide.
Oh, sorry, I'm on the wrong.
Okay, so I'm going to stop there for the moment, but I want to encourage everyone to think about participating in CHAMP2, which we hope to launch even as early as later this year, to ask ladies to come back to now have 2 points to look at them, right? We have the baseline, but we'd like to have a second point to see what changes. Julie?
Do you want to say anything about your people?
Oh, this is my slide. Sorry.
Yes, it's you.
I want to acknowledge a vast number of people at Vanderbilt that have been involved in maintaining and completing this study over many years. I particularly want to thank Dr. Alexander Conley, who's been a stalwart colleague throughout the 6 or 7 years of this project, both from conception to execution. We couldn't have done this without you, Alex. Even as you go off soon to back to Australia, we look forward to continue to work with you. I want to also acknowledge Laura Mendenhall, who's jumped in the last year to be our project coordinator, who's done a great job. Sarah Nelson, our nurse practitioner, has been amazing. Jason Russell is not on the call, but has done a lot of our brain imaging analysis, and the many other colleagues and professionals who've played an amazing role in this huge project.
My turn. Alrighty, I just want to do the same thing for the folks at the University of Vermont. I think I saw that Jana Markarowitz is here. She's the research specialist coordinator extraordinaire, does everything from the recruitment to the MRI studies and analysis. And I couldn't do any of this without her. And I think folks really enjoy her. Olivia Knopf, I think, might have a PhD by now. She went off to grad school, and many folks work with her in the pandemic days. A lot of physician colleagues at UVM. Since I'm not a physician, I have support from so many of my colleagues, including Jim Boyd, who did all the spinal taps. I also want to say a special thank you to the Clinical Research Center nurses, the CRC nurses. They are, again, it's a great facility, and we are a nice, well-kept secret in the hospital, and happy to work with you folks on this work. And I think that's all. And we just want to say thank you. Thanks for listening. Thanks for coming. Thanks for being a part of all of this. And we hope we can convince you to come back soon. I'm going to stop sharing, and so we can see if we want, if you'd like to ask some questions, you can I see a question, I see a hand. You can put your real hands up, you can put your Zoom hands up, you can write a question in the chat, and we will do our best to find you and answer your questions as we can see them. So take it away, Eileen.
Thank you so much for this, first of all, and thank you for sharing with us. It's been quite a journey, and I'm just so delighted that you are where you are. And I'm very delighted to hear that you are continuing this, because that was going to be one of my questions, is you have such a wealth of information here. Follow us, see what happens to us over time. But my other question, the real question is, so I've recently started HRT. And I see that exempts me from a lot of the future work, at least what's going on right now. But I'm wondering if you might want to consider actually including people in the study. So originally I wasn't on it. I am now. That might be some interesting information for you to think about. What does happen with some, because you have real-time data from pre-HRT, and then you could perhaps see what's going on while you have that. I won't be on it forever, but there's probably those of us that have that profile, and as you're trying to understand what things are effective down the road, you could have some real life data to try to examine. So just throwing that out there.
You want to go first, Paul?
I think that's a great idea, Eileen. I think that we do need to understand what happens when HT is added. And so I think the answer is yes. We will want to work with you and other women in your situation to follow you up regardless of what your hormone status is, because we have this, as you pointed out, we have this amazing, unprecedented depth of data on everyone, and understanding what happens next is a critical variable. We have learned so much over the past 25 years from large scale cohort studies that have followed people for many years, and I think the impact of HT is still really not well understood long term. And so I think this presents a unique opportunity to ask those questions.
And I'll just add, I think experimentally, it's interesting. No matter what is happening to folks, we want to bring them back. So as you mentioned, Eileen, we do have all these measures, and we want to see how time and whatever else has changed. In terms of how would we design a new study? Am I going to do a new study with 50-year-old women? I think the world is changing a little bit, and so we always like to have these really specific enrollment criteria with healthy people. But then we don't find the interesting stuff. That's not interesting. That is interesting too, but we don't find all the stuff, So I think, certainly and Paul and I were doing this work in the early 2000s when the WHI studies were stopped and everyone stopped taking the HRT. And now the pendulum is swinging back and people are coming back to it. The recommendations are back to it. So, we're back to the early 2000s. So I think it's a good question. And I think we'll be able to study it now because more people are taking HRT also.
That's right. We used to joke, Eileen, that estrogen stopped working in the year 2000, but now it's working again. We
Thank
had a
you.
question from Susan, I believe.
Yes.
What is the relationship or correlation between p-tau 217 and ApoE4? So these measure two different things. So p-tau 217 is a protein that's measured in the blood or spinal fluid. ApoE4 is a genetic marker. It's a marker of a gene that we all have called ApoE, and each of us has one or more variants of that gene, either 2, 3, or 4. The 4 is the higher risk variant. And so what we think is the likely correlation is that people who have one or particularly 2 copies of ApoE4 will likely have a higher risk of having higher levels of p-tau 217 as they age. So p-tau 217 is not a diagnosis of Alzheimer's disease. It is simply indicating that the brain may be accumulating amyloid. We all will have a certain level of p-tau 217 in our blood. But if it starts to climb, then that may indicate that there's higher levels of this amyloid protein being deposited in the brain. So there's no direct correlation between these two, but the genetic, the ApoE4 gene version is considered higher risk to have an elevated p-tau 217 as one ages. Doesn't necessarily mean it, but it just means higher risk. I hope that is helpful.
Thumbs up. Barbara?
I'm trying to type this out and I'm not quite sure how to word it. So when you were You were discussing the slides about, oh, this kind of surprised us. Like, we were not using the placebo, and there were some people that were actually doing better. Do you have any like preliminary predictions about what that might be?
I know we like to call it compensation. Like, so we think that it's like you're getting this challenge. It's like you can think of a number of different, like an exercise challenge or a cognitive challenge, and there's something that's harder, and so you're working harder to solve it. Like, as you know, you folks have all been on this medication. You're like, I don't feel sharp. I don't feel as good as I normally do, and I'm not sure that feeling is even that obvious. But something's different. I'm gonna try really hard because I know these scientists are want me to do well, or whatever the motivation is, and you may not even have the that sort of explicit ability to change it. But we're thinking that there's something people are having this challenge with this medication sitting on your receptors, stopping cholinergic systems, and there's some, whether it's a top down, like I'm thinking I'm going to try harder, or it's some other physiologic process that like we don't even know yet, that people are somehow able to do it better, which does, I mean, it's not like we've seen it in a number of different studies over a number of different years, and we haven't quite dug into it because it's sort of I don't it's weird. Paul, what do you think about that?
Well, I do think that compensation is part of the answer. And I think it's well to remember that there's a distribution of compensation. One of the things that we have understood over the years is that changes in brain activity can't be thought of as a sort of linear construct, right? It doesn't mean more is better and less is worse. Sometimes it can be the opposite, actually. It depends on efficiency or how the brain moves activity around to compensate. So at this point, we're not entirely sure of our interpretation of this, but I do agree that I think it's a compensatory response. And what I'm interested in, particularly as Dr. Dumas outlined earlier, is I want to look at people who show clear compensation versus people who show less compensation, because I think that may have something to do with their long-term risk for cognitive impairment as they age, or changes in cognition as they age. That's a hypothesis. We need to prove that. It's
Just
not established, but that's where we're going.
did the MRIs show any like different lit up areas, like different pathways that people were using?
I mean, we look at them overall on an average, for the most part. So yes, there's definitely individual differences, like people, on average, and then when you look at the average, you you get rid of all the individual differences. So, especially with the imaging and the activation stuff, it's a little bit, it's hard to do individual analysis, with that because it's better in a group average. But we will, as Dr. Newhouse was saying, we will try to look at this thing where maybe if we separate, maybe we have some folks who are real compensators and folks who aren't as doing as much of the compensation. Let's see what the brain activation looks like. And we haven't done that yet, but that's sort of one of the next steps.
Thank you.
See some questions in the chat. Do you see those, Paul? Current age cutoff or criteria for HRT? Do you know? I don't know that answer.
Yes, I do not know the answer for that. Not being a gynecologist, I'm really not the best position to answer that.
I
But
can answer that. Actually, it's actually not an age cutoff. It's more about how long it's been since you had your last menstrual cycle and your symptoms. And so they put together your individual profile and then make a determination. So I don't think there's an actual cutoff per se.
I think that's probably accurate. Yes. All right, very good. Let's see. We have a question. What if some of the participants are beginning to experience cognitive decline? Are you interested in hearing from those participants? Absolutely. The answer would be we are particularly interested in following up with those individuals. Also, Dr. Albert's study is particularly relevant for those individuals. If you're in the Nashville area, particularly, that would be an extraordinary opportunity, and I think she would She's working very closely with us and recruiting participants, particularly from our CHAMP group of ladies. And so I think she would be very interested in hearing from you. Let's see, do we have any other questions that we can answer for people? I know it's late in the day for some of you.
Here's one.
What if someone has a high 0.26 p-tau 217, but no cognitive issues yet? Yeah, that's a great question. So that is a question we are actually trying to approach on a national level. So there is a large study that we're one of the sites for here at Vanderbilt, which is attempting to take individuals who have elevated p-tau 217 and also have a confirmed high brain amyloid, or elevated brain amyloid, I should say, and put them on a prevention treatment to see if it will essentially stop or reduce the risk of them ever developing significant cognitive impairment. And so that's called the AHEAD study. It's currently closed to enrollment right now, and it will read out in 2027 to 2028. Unfortunately, it won't read out till then. So we won't know whether this strategy works, but I think we are extremely interested as a field in taking individuals who do have elevated biomarkers like p-tau 217 and randomizing them to prevention trials, or offering them the opportunity to be in prevention trials. I think you're going to see more of these come along. We would be obviously very interested in following you. Currently, there's no available FDA approved treatment for people that have no symptoms at this point. And so there is a debate within our field about whether these people have Alzheimer's disease. Do they really have it, or are they simply at risk for it? Kind of like high cholesterol. Are you at risk for heart disease, or do you have heart disease? We think you're at risk for it, but don't necessarily have the disease. And so reach out to us, stay in contact with us. We'd love to continue to have you work with us as part of the successor to CHAMP. How much longer will you be analyzing?
It's a whole career worth of data. We'll never stop.
Julie, you're probably best able to answer
I'm never
that.
stopping. I don't know.
I'm a lot closer to retirement than you are.
Right. I hope this is my retirement project. There's a lot we will not, there's always more questions. So we haven't written the main paper yet, so that'll be the first thing to do.
Yes. So I think the answer is we're going to be analyzing this data for at least the next year or two. We'll have more to be able to tell you about as we get all of this data crunched. As you can imagine, it's an enormous set of data. I mean, I always like to quote the Bible, right? The harvest is rich and the laborers are few. And so we have a rich harvest of information, and we're just starting to wade through it. Is it true that if significant amyloid showed up in the brain, that participants were contacted? So if not contacted, no significant amyloid was seen. That is generally true. So if we identified highly elevated brain amyloid, and we did in 2 or 3 people, I believe, we did notify you and invited you, particularly if you were at Vanderbilt, to consider participating in the AHEAD trial, which a couple of people did. And so because we felt that was sort of actionable information. So if you weren't contacted, at least at the time we saw you and you did your initial amyloid PET scan, we did not see a clinically meaningful elevation in amyloid. Now, as they say on Wall Street, past performance does not guarantee future results. So I can't tell you what would happen a couple of years down the line, but at least at the time we didn't see it if you weren't contacted. All right. we thank you, and we thank you, all of you, for being the most amazing people for participating in these studies. And you know, I used to joke with Dr. Dumas that the motto of our lab is that we do nasty things to nice ladies. And but it's all in the service of understanding
Yes.
the risk that we all have for Alzheimer's disease, and why women are overrepresented in this process. And we need to make progress on this, that's for sure.
All right.
Well,
I
we've
just want to
reached
add
the
my
end of
thank
the
you.
hour. Yeah.
Yes, we're at the hour. Let's see, just more thank yous in the chat. And I just want to add my thanks to everybody for, again, for your participation, for your questions, for your engagement, and we hope you'll answer our emails when we we get back in touch soon. Um, so thank you very much.
All right. Take care. Have a wonderful evening, everybody. And feel free to reach out. I'm sure to Dr. Dumas would be interested in receiving any emails or questions.
Absolutely.
And I'm happy to also receive any questions or emails or contact. And so do stay, as Garrison Keillor used to say, be well, do good work, and keep in touch.
All right. Thank you.
Bye-bye now.
See ya.